Hello.
Hello, thank you for joining us. We are just about to get started. So as a reminder, all Genetic Society of America online events are covered by a code of conduct, and the link for that will be available in the chat shortly for anyone who needs to take a look. I will go ahead and turn us over to Dr. Hugo Bellin. Hugo, you are good to go.
Hello, everybody. Welcome to this second seminar in the series. And today, Nara and Michael will be talking about matchmaking interests across research organisms. And first, a brief introduction. Michael will be the first speaker and will then relay to Nara. And so Michael did his BS at UCSD and his MS as well. And he worked in the lab of Ethan Beer and got an interest in fly biology there. And he's still interested in fly biology. He got an MD at Baylor College of Medicine and in pediatrics and focused on medical genetics. He then became an assistant professor in the molecular and human genetics department and has been focusing on human disease discovery and is part of the Undiagnosed Diseases Network, the Center for Mendelian Genomics, and is playing an important role in the Model Organism Screening Center. With respect to Nara, a very different CV, Nara Sobreira. She got an MD from Brazil. In clinic, then she also moved on to clinical genetics in Sao Paulo. Were you related to Mayana Zatz at some point? No. She then decided to do a PhD after her MD degree and moved to Johns Hopkins, where she stayed ever since. She did a postdoc in Johns Hopkins with Dr. Valli, and then did a residency and became an assistant professor at Johns Hopkins. And her interests have been also in human genetics. She's currently, as I said, an assistant professor, and she's focused on databases in PhenoDB, GeneMatcher, which she'll talk about today. And she's also part of OMIM. And so welcome, Michael. I think you're starting off.
Yes.
Please share your screen.
Okay. All right. Thank you for the introduction, Hugo. And it's a pleasure to present to you all and also to present alongside Nara and hear about the work on GeneMatcher. So, the focus of the session today is meant to discuss sort of matching interests across research teams. So why is, matching interest more productive than ever? So the argument is, of course, collaboration has always been a crucial activity for scientists, but in the current environment, there's really never been a better time to match across very diverse fields. So recent developments in genomics, and then of course in like global communications, have basically allowed very rapid transfer of genomic knowledge from real patients who are in the clinic seeking diagnosis to basic science labs and then back. And this is sort of easier and faster than it's ever been. So we're going to talk about matchmaking in this session. And I wanted to sort of bring up a couple of potential definitions of matchmaking that we will be exploring both of these definitions. So matchmaking, could be thought of as a process where we essentially compare the human phenotypes for individuals who have variants in the same gene. For example, an individual may be seeking a diagnosis, they have a variant in a candidate gene. We find other individuals with variants in the same gene and we compare the phenotypes. But we'll also discuss matchmaking in this session and in the context of, particularly members of the Genetic Society as a process where we match a body of research or an area of expertise in a particular gene or genetic pathway with, patients who are carrying variants in that gene. And hopefully the two tools that we'll focus on will kind of explore both of those senses of matchmaking. So I was going to start off with real world examples. These are examples that I happen to be particularly familiar with that I hope will illustrate those kind of two definitions. So I'm going to start off by talking about a gene called NACC1 and several families that we had experience with that really illustrate how one nucleotide in the human genome can sort of be matched across several families and lead to diagnosis. And then I will talk about a patient and a family of their daughter Avery, who has a disorder that we're able to study and understand some of the aspects of the variant in model organisms. So for the first example that I wanted to tell you about, this started actually with a patient who presented to the Undiagnosed Diseases Network. And this was a family who were seeking a answer for their son who had suffered from microcephaly, some cataracts, and a number of neurodevelopmental abnormalities with epilepsy. And upon presenting to the Undiagnosed Diseases Network, a candidate variant was identified in the NACC1 gene. And when this project started, there was essentially the one patient with a de novo variant in this one gene. But of course, it's an N of 1 observation. But through being in the Undiagnosed Diseases Network and interacting with other genomic research studies, the first patient was initially connected through GeneMatcher to a patient at Children's Hospital of Philadelphia with some phenotypic similarities. Then a third patient was identified in the Centers for Mendelian Genomics with cataracts, epilepsy, intellectual disability, and then two more patients. And the point here is that went from one individual N of 1 observation with one candidate gene to 5 individuals with the same gene within about a 3-week time period through this gene matching effort. And ultimately, in the original sort of initial group that was identified, 7 individuals with a very similar phenotype sharing cataracts, epilepsy, and some neurodevelopmental abnormalities were all found. And significantly, all the individuals were found to have an identical de novo variant in the same nucleotide of the NACC1 gene. Interestingly, one of the individuals actually was somatic mosaic for that change. So the point here is that within a very rapid time period, went from one individual to a cohort of individuals, all of which sharing the same nucleotide change, all occurring de novo, so not present in either parents and found in the affected proband in those families. So the lesson for this story, of which there have been many others in human genetics literature, is that when we use genomics and we can rapidly communicate across teams, we can make discoveries and diagnose patients very rapidly. And this is particularly powerful for cases involving de novo mutations. And significantly, I wanted to point out that, the initial patient I brought up, their diagnosis sort of came from the discovery of the other patients. But in our role in the story, we actually had patients who had been seen in our hospital with a mutation in this gene that were also solved. So all the families sort of received this answer simultaneously. So this gene matching effort, which Nara will cover in the GeneMatcher tool discussion, is just particularly powerful, particularly powerful tool. The second story that I was going to go over pertains to a patient and a family that became very special to us at Texas Children's Hospital. So this was a patient who was born after an uncomplicated pregnancy, and there were concerns about developmental delay that started at about 4 months of age. The patient had a motor incoordination, impairment in communication, particularly in expressive language. And by the age of 8 had been enrolled in the Undiagnosed Diseases Network, and also the Centers for Mendelian Genomics after a very significant diagnostic odyssey in the clinic trying to find the cause of her symptoms. So this shows that the MRI had shown a progressive change in her cerebellum that went along with sort of clinical ataxia. And an exome reanalysis for this patient through those studies actually identified a de novo change in a known, better characterized disease gene, CACNA1A. And there were also 4 other subjects studied that had a nearby de novo change. So this is showing kind of what was known about the area of the protein where our patient's mutation was found. And what's significant here is that it's within the 4th transmembrane domain. And within this S4 segment, we knew that there were instances of episodic ataxia, spinocerebellar ataxias, and familial hemiplegic migraine, all due to mutations in this same segment. But our patient's particular change had not been seen. So in this study, we actually designed genomic constructs for the same missense variants because they were conserved within the Drosophila genome as well. And we're able to then study those genomic constructs using cacophony mutants, a well-known gene within Drosophila, the homologue of the CACNA1A. And interestingly, we, to summarize some work on this gene, we found that while neither mutation had the ability to, rescue, say, cacophony phenotypes, we saw a unique sort of neurodegenerative phenotype that came from our patient's mutation. So what's shown here, the Drosophila photoretinal photoreceptors, and what you can see is below are the 2 patient mutations and our patient's mutation shown here on the right, showing a loss of a lot of the photoreceptors at a fairly early time point. So this patient's story sort of has continued, and research into this gene has really, even though when this patient was found, a lot was known about CACNA1A, a lot was known about ataxias and familial hemiplegic migraine, but this sort of opened some new avenues for studying model organism studies of these unique novel mutations that haven't been seen before. And over time, a number of new patients with an expanded phenotype have come out for CACNA1A. There's also a family-centered foundation currently supporting research, particularly into epilepsy phenotypes for this gene. So kind of going to show that model organisms can help reveal, even for genes that we think we understand in humans pretty well, new functional aspects. So I'm hoping that these first illustrations give you a little bit of excitement about matchmaking as an effort. And the arguments that I would like to make is that matchmaking is important for several reasons. So first of all, there's obvious medical advancement that can happen from this matchmaking. We have patients that get a diagnosis that they didn't previously have in the rare disease community at a very rapid rate from matchmaking. We also get better knowledge of gene function that when we look forward to new therapies such as gene therapy or antisense therapy, we really need to know, are these mutations loss of function? Are they gain of function? How do they affect the gene? I would also argue that science itself benefits from understanding rare Mendelian disease And rare disease offers us insight into common disorders because it reveals pathways that are at work in the human body and also can show us basic biology that may be new and unexplored. So, and this is just kind of summarizing that scientists play very crucial roles in the process of rare disease research. So starting with, if we think about rare diseases with the patient and the family, they may undergo a diagnostic odyssey. They would then hope for a gene discovery, and then there would be potentially a therapeutic odyssey from that point, going from the gene to a treatment. And particularly model systems play important roles in several steps here, both in shortening or ending the diagnostic odyssey, bringing to a gene discovery, and then also in understanding gene function and helping guide therapeutic ideas. So, so hopefully giving some enthusiasm for matchmaking as a activity, we're going to now talk and focus on two tools that will hopefully continue to illustrate the two ideas of matchmaking, matching human phenotypes and then also matching models to genes. So I'm going to start by switching over to Nara, who will cover GeneMatcher at this point.
Good afternoon, everybody. Thank you, Michael, and thank you, Hugo, for the invite to talk here today in the meeting. So let me make more presentation mode here. So I'm going to talk about data sharing using GeneMatcher, and later towards the end, when we have 5 more minutes, I'll talk to you about a new tool that we developed based on the experience using GeneMatcher. So how did it start? We all know that 10 years ago, whole exome sequencing and most recently, whole genome sequencing started being used for the identification of novel disease genes for patients with rare Mendelian diseases. And the use of these techniques generated a big amount of data, a large amount of data that needs to be analyzed and has been used for the discovery of novel disease genes and novel disease variants. This is to show you how that has actually helped us to discover new disease genes relationships in the last 10 years. You can see here, this graph comes from the data in OMIM, and the purple bars are the number of phenotypes that have been added to OMIM in the last 10 years or so. And the orange bars are the number of genes. About 200 novel phen-gene relationships are added to OMIM per year. And you can see here that the number of genes that are known to cause disease phenotype has gone from 2,500 to 4,500 in the last 11 years. And this is mostly due to the use of whole exome sequencing and some whole genome sequencing most lately. So how do we go from this large amount of data that is being generated to the discovery of genes and variants that are causative of diseases? There are many steps that go into that process, but one step that became clearly very important and necessary, like Michael was just showing you, was data sharing, right? So it became clearly apparent that we needed to share the data to be able to find the answers for our patients. So how to share this data? That the conversation started as a long time ago, even before whole exome sequencing, because people are doing arrays. We're already talking about data sharing. Decipher is data sharing, and it's much older than any of these projects that we are talking here today. But when we started talking about the data sharing of the whole exome sequencing data, many questions came up. And here's just some of the questions that we had when we were talking about it. What kind of data to share? Gene names, phenotype, variant level, raw data. So all these questions about what kind of data would be important to share, how to share, where to share, with whom to share, who should be giving the data to, and what kind of data to whom. And then the problem with data sharing at the beginning was, how do I keep the power to actually own the publication or the results of the data sharing, the results of the findings? Who is going to be the first, the last author of the paper? So that sounds like it's not important at all, but it was a main thing that we had to overcome when we're talking about data sharing. So it was a very important piece of the conversation, actually. And then, of course, how can we share data and still keep the privacy of our patient and follow the IRB rules of our institutions? That was another thing that was very important when this conversation started about data sharing of the whole exome sequence. These are just some of the questions and some of the issues that we had to discuss. It's not all of them, just to give you an idea of how complex it was, and it still is. So in nowadays, we have many different levels of data sharing, and it goes from databases that are NIH funded and NIH supported to share the whole raw data that is generated with NIH funds. So I have here the dbGaP, NView, Kavarica. These are all places where you can go as a researcher, make your subscription, your registration, and try to get raw data from a project to do your own analysis. Very complicated, not for just anybody to do it, but it is possible to get this data and do the analysis. And then the public level of data sharing, that is de-identified data at the variant level, mainly with information about frequency and also variant classification, how these variants were classified by research labs or clinical labs. This is all very important for us when we're analyzing data, and it's available in many different databases. Like I listed here some of them, not all of them. These are the ones that I'm most familiar with, but many of you may know some others that are not listed here. The one problem here is phenotype. Very few of these databases that are publicly available have phenotype information available. And it, like Michael was showing, phenotype, it's a very important piece of trying to address if the gene is pathogenic or not. So what was missing? Phenotype. Most of these databases don't have phenotype information. Decipher is one of the few that you get phenotype information on the patients that are described, or the variants and genes that are described there. In the case of Decipher, mostly CNVs, copy number variations. Family history, right? So family history is basically not at all available in any of these databases. And also the capability to reassess the patient. The one problem with description of phenotype that we know of is that when we describe the phenotype is not completely described. Like it depends on what the patient has been doing in the last few years in terms of follow-ups and how he was actually assessed and what kind of test he has done. But sometimes when we say like, oh, the patient doesn't have a heart anomaly, the patient never had an echo. So we don't really know if he has a mild heart anomaly that's not there. And if I don't know who the patient is and I cannot go back to actually reassess, I will never know if that patient had or not a cardiac anomaly. So these are some of the things that are missing. And these are some of the things that we try to assess, to cover when we were coming up with our idea of how to share the data that we had. So as Michael said, we are actually part of the Baylor Hopkins Center for Mendelian Genomics, and we are generating a lot of whole exome sequencing from many patients with rare Mendelian disease. And we had this data that was coming out of our analysis, and we wanted to share to be able to actually solve our cases. And then we decided we are thinking of how to do it, and we decided to focus on few things. We were going to share as little as possible, so we would have as little as possible problems with our IRB and patient privacy issues, but we wanted to focus on what we thought was essential and more specific. And in that case was to start with the gene name. Thought like, okay, if I come up with a small list of candidate genes that are strong candidate genes, and I share these genes, and I can find other people who have patients with mutation in these genes, I can go from there to find out more about the phenotype. And to avoid the issue that people were talking about, like, but I'm going to be scooped if I put my genes in there and someone just sees that I have a patient with these genes, but doesn't tell me and just go around me and does whatever they need to do to publish. We said, okay, we have to generate a process or create a process that notifies everybody who is involved with that gene at the same time, who they are and what's the gene that they are interested. And then nobody will know of the information first, like everybody will know the information at the same time. So this is when we created GeneMatcher. So GeneMatcher was created in 2013, and it had in mind to cover all these issues that we talked about, but not perfectly. There is no data sharing database that is perfect. We all do slightly different to try to cover all the issues, but what GeneMatcher decided to focus was like, we wanted to connect patients, families, clinicians, researchers, anybody, not only human researchers, model researchers, that had an interest in the same gene. Okay, so if we can connect them and they can talk to each other on this gene name that is de-identified data, right? I'm not talking about, I cannot identify anybody if I say that I'm interested on gene X. This is all. And the matching would be automated. Everybody who was interested on that gene would know about it at the same time, about the others. And they would choose from there to contact each other and share more information. So I just got an email saying that Mike is interested on gene X, and Mike just got the email saying that Nara is interested in the same gene X. Now, if I don't like Mike, I'm just going to say, I'm not going to send him my email. This is all, bye. But I think that rarely happens. And most of the times people just decide to connect to each other and say, hi, Michael, I saw that you have interest on this gene X. What's the variant that you have on your patient? What's the phenotype that your patient has? And I tell the same information about my patient. You do have the option of actually already including that information or part of that information in the entry when you create an entry related to a gene, and that information already goes automatically on the email notification telling about the match. But if you don't want to put that, you don't have to. The only thing you need to put is a gene name. Later, after we started matching on genes, we also decided that we would allow matching on phenotypic features, genomic location for variant level, and on mean disease number. So these are also available in GeneMatcher. They're less used, but they are available. How is GeneMatcher doing now? So as of July 1st, we had 13,700 genes in GeneMatcher that had been submitted by 11,800 submitters from 93 countries, and 8,600 of these genes have matched. So it's about 63% of the genes that are in GeneMatcher have had a match. Who are the users? Almost 8,000 of these 11,000 users, almost 12,000 users, say that they are researchers. 5,000 of them say that they are healthcare providers, and 635 of them say they are patients. We don't really do a lot of advertisement for patients. We are trying to increase the advertisement for patients so they can use more GeneMatcher, but they still learn about it and they still come and use it. You do have an option when you create your account to say if you don't want to match to any level of submitter. And if you say, I don't want to match to a patient, you will not be matched to a patient and you will not get an email notification of a match with a patient, and the patient will not get your information also. So that's possible for the case of researchers or any other user that doesn't feel comfortable talking to patients directly. Who are the model organism users? So we have 323 genes that have been submitted by model organism researchers. It's not a lot, as you see. We have 13,000 genes in there, and only 300 of them are from other model organisms that are not humans. But we have been trying to increase that relationship with the model organism field, and one of the ways we are trying to do that is connecting with ModelMatcher that Michael is going to talk to you later. But here are the model organisms that are in GeneMatcher. Most of them are mouse models, then followed by zebrafish, followed by fruit fly, mostly from Hugo's lab and Michael work. And then some from yeast, and then a few others from different model organisms. So, but they are very much welcome. And actually, the first paper that acknowledged the use of GeneMatcher was a paper that matched a patient model with a mouse model. And that was made possible to define that this gene was actually a pathogenic gene, a disease-causing gene. So, oops. What are the genes that are in GeneMatcher? Just to let you know a little bit of what's in there. GeneMatcher is mostly to try to find a disease to a gene that has not been associated with a disease before. But sometimes different diseases are caused by the same gene. So people actually put genes that are already known to cause a disease because they're trying to find sometimes a novel phenotype associated with this gene. You see here that 3,000 of the genes that are in GeneMatcher actually are associated with a homing phenotype already. But 4,730 of them, and then 4,730 of the genes have never matched. As I told you, like 63% of them matched, but almost 40% of them didn't. And the ones that didn't, 3,873 are not in homing. So, and the ones that, and some of these genes have been submitted many times, like 2,800, almost 3,000 of them have been submitted more than 5 times, and 1,800 of them are not in homing. So we actually just submitted a paper doing an analysis of these genes, a more detailed analysis of these genes that have not matched and that have matched so many times. What do they look like in terms of like constraint scores? PLI score, missense Z-score, RVIS scores. Why are these genes matching so many times and some of them are not at all? So we just submitted the paper. Actually, it's almost about to be published on more details about these genes. And I don't have time to go over that here, but just to let you know, it's coming out and you can, if you're curious about it, you'll be able to read about it soon. So how is GeneMatcher helping? More than 484 publications have acknowledged GeneMatcher. As of June 12, we took a look at these publications a little bit more closely, and 302 genes were described in 249 publications. 157 of these genes were associated with autosomal dominant phenotype. Michael was just saying how it works well for de novo variants, but it also works very well for autosomal recessive phenotypes. 105 of them were associated with autosomal recessive phenotypes, and then 24 with both, and 6 were X-linked recessive and dominant. I think it's because actually we have very few genes that are X-linked in GeneMatcher. 221 of these 302 genes had been GeneMatcher for more than a year before the publication. And 231 of these 302 are now associated to a phenotype in homing. 73 of them actually had a homing phenotype before the gene was posted in GeneMatcher, and still there was a match and there was a publication after that match. We do think that it's more than 484 publications that actually made use of GeneMatcher information, but it's very hard for us to track. The only way we can track is if the paper acknowledge GeneMatcher. So, but by looking at the genes that had matched more than 5 times and the time that they had been in GeneMatcher and the time that they become a homing phenotype, it sounds like when we had these 302, actually 445 genes could be maybe linked to GeneMatcher matches. So what else GeneMatcher is linked to? As we said before, when we created GeneMatcher, a few other databases were also being created, like Phenome Central was another one that was coming out when we were creating GeneMatcher, and we decided that we didn't want users needing to create an account in GeneMatcher and then an account in Phenome Central. We wanted them to go to one place and from there be able to go to others. And we created what's called Matchmaker Exchange. We created an API that would connect databases that are intended to work this way that we just talked about. It was the first connection was GeneMatcher and Phenome Central, but then we soon Decipher joined, and then MyGene2, and then we have now Seeker, IRUD, RD Connect, and Patient Matcher. And soon, hopefully, we will have ModelMatcher. We are now talking about the details of the connecting them to the other nodes also. So you go to one of these databases, and from there, you can send your information to the others and match to other users that are in these other databases that are in other countries many times. So Phenome Central is Canada, MyGene2 and Seeker are US-based, but IRD is Japan, RD Connect is Europe, Decipher is Europe, and Patient Matcher is Europe, all in different countries in Europe. So this is where we are now. I hope I didn't take much longer than I was supposed to. And I will save our next step to the last 5 minutes at the end. Let me stop sharing my screen here. Did I stop sharing? No, I didn't. Here it is.
Great. Okay, great. Thank you, Nara. So, all right. So then I will now go over the ModelMatcher. So as Nara pointed out, the GeneMatcher, does allow model organism researchers to, match with genes across, potentially human genetics groups. The idea behind ModelMatcher was to provide the model organism researchers a tool that was, synergistic and wouldn't compete with GeneMatcher. There's really no reason at this point not to, use both tools. The idea you'll see going forward is to integrate, ultimately both tools. But the idea was to really try to bring in more of the model organism researchers and to allow things like matching across models. So ModelMatcher is live. It's shown here, ModelMatcher. Remember, it's .net, not .com. And ModelMatcher is the work particularly of Mike Harnish, a PhD student and or a PhD candidate at this point, and in Shinya Yamamoto's lab, with a lot of programming help from Zhendong Liu's group and a lot of insight from the RDMM project from Canada. Oops, I'm sorry. So the idea behind ModelMatcher is, if you think about a model organism researcher starting with kind of a set of genes of interest, and the idea is for them to register that interest into the system of ModelMatcher. And then the hope is that that could integrate them or allow them to interface with clinicians who may have undiagnosed cases with candidate variants in the gene of interest, or maybe there would be the gene matching diagnostic odyssey is not as much the focus. Maybe what's needed is mechanistic or therapeutic research, and so that's another area where the hope is ModelMatcher could help integrate a basic science lab. Also, the idea is for other stakeholders, patient, parent organizations who may not necessarily easily find researchers who've spent long years working on their gene of interest, to allow those communications to more rapidly occur. So the initial steps for ModelMatcher involved integration with the current infrastructure at work in Canada, particularly spearheaded by Canadian group for the Rare Disease Models and Mechanisms Network, but also at use in Australia, Europe, and Japan. And then going forward, the idea is to, have sort of integration of ModelMatcher with the Matchmaker Exchange and GeneMatcher, as Nara mentioned. So that sort of an idea of an overview of how this would work is you could start out by thinking about a scientist who says, I would like to register interest in my genes within ModelMatcher. And they would basically provide name, contact, gene, and then the sort of types of models available. Then the idea is that clinicians or other stakeholders, patient advocates, could go and ask, is there a researcher working on this candidate gene? And by registering, similar contact, it's again an idea to facilitate ultimately collaboration. So one little difference that might, that is thought to be useful is that there's kind of a search and match feature to this. So, for instance, a researcher could generate a list of genes that they, have extensive expertise and publications on, where they have a lot of track record and confidence about those genes. And those could be available for, say, like a public search that anyone could go into ModelMatcher and do a rapid search for those genes. But then there also may be a place for, more direct collaborations or genes that are more in development. And here the match feature could come in, where someone could register interest in a gene sort of in a way that would facilitate direct emails and matching. And this would work similarly to the way that Nara presented GeneMatcher to work. The only difference might be is that potentially you could have a fly group connect with a worm group over a gene or pathway of interest. So the hope around these tools is really to help to integrate the scientific research community and the clinical research community with the rare disease patient and family community. So the idea is to really, help scientific research be more integral to the process of rare disease, and then for clinicians to be able to come in and provide candidate genes and find researchers more quickly, and then for patient advocates and families looking for diagnosis and treatment to be able to interface more quickly with this community. So I think we're going to end this session by going over a couple of new aspects and lessons. So we're going to start by talking about matchmaking and the variant matcher interface. And then at the end, I will go over some lessons of communicating across teams that we've learned, particularly from the Model Organism Screening Center for the Undiagnosed Diseases Network. So we'll go now to the variant matcher.
Unmute and share my screen. So, all right. So once we were sharing our genes that were candidates after the analysis of whole exome sequencing, and after GeneMatcher was being used by many different countries, we kept getting emails about, okay, but do you have a patient with variant X or variant Y or Z? Because the candidate gene list, it's a very small slice of the whole exome sequencing results of an unsolved case, right? This is what we, with all our biases, select as the best candidates. Doesn't mean that we are right, and still we have hundreds of other variants that could be in there. But we are not going to share hundreds of genes because it would be very unspecific. We could share the variants, because if we are trying to match on variants, then it's more specific and less likely to be a false positive if a phenotype also is a match. So we created Variant Matcher to share the data of the Baylor Hopkins Center for Mendelian Genomics at the variant level. So basically, this is what we had sequenced with the Baylor Hopkins project. We had about 9,000 families that had been enrolled in the project, and about 8,000 of them had been sequenced, and we had genomic data from these patients, mostly whole exome sequencing. And we had made many discoveries, but we still had many cases that were unsolved. So we decided to put this data out there to be queried and try to solve some of the cases that we have in our project that are unsolved. This is a little bit different from GeneMatcher. GeneMatcher allows anybody to put their own data in there. In Variant Matcher, it's only our data that we are making available to other people around the world to query. But we are not allowing them to deposit the data in there to be queried by other people also. Not at this point, at least. Maybe in the future, it is something that we are going to be doing. But as of July 1st, we had genomic data from 6,200 individuals, and these come up to be almost 900,000 unique variants. These are variants that are rare with a minor allele frequency lower than 1%, and variants that are coding, not including the synonymous. If I splicing, it's also predicted to affect, if a variant is intronic, but it's predicted to affect splicing, we also allow it to be queried. And it's only at this point, single nucleotide variants, not the indels yet. We are also working on changing that. But the way it works is like you create your account, you are approved by an administrative committee, and then you can search up to 10 variants per day. Our data, it's on HG 37, but you can put your variant on 36 or 38 and you will lift over to 37 to be able to match in the database. We don't match on features, but you have the choice to put the features of your patient here. And if there is a match, the features will be shared on the email notification. The features of your patient and the features of the patient that you matched with in our database. You put your data in here and you query the database today. If in a month from now we had added more individuals in the database, your data was not saved, you are not going to know if there was a match. So you have the option to save your search, and then your search will be redone monthly. And if there is new data in there and now you match to someone in the database, you will get a notification about that. So this is how it looks when you save your variant for continuously be matching month after month. So as of now, this is new. We started actually up and running about a year ago. As of June 1st, we have 671 submitters from 44 countries, and we have 70 variants that matched to 822 individuals in our database. What happened to these matches? When we had 61 variants that matched, we took a closer look at them. They were in 56 genes, 40 were in non-disease genes. So most of these people were trying to actually classify a variant of uncertain significance in a non-disease gene. They were mostly rare variants with a minor allele frequency lower than 0.1%. And what happened most of the time was the variant could be classified as benign because there was a match to many individuals in our database that were some of them affected, some of them unaffected, and the ones that were affected had many different features, phenotypes. So really the variant was most likely benign because people didn't share at all the same features that were, the people who were matching on this variant didn't share the features. But every time that happens, we still tell people to be careful about ruling out that as a positive match because we have to take in consideration incomplete penetrance, variable expressivity of the phenotype, age of onset of the phenotype. So these things need to be taken in consideration before you say like, oh, the patients don't have the same features. This is not a match. The variant's not causing the phenotype on my patient. So this was what I wanted to say now towards the end, Michael, and thank the group who has been working with us on all these projects throughout the last 10 years. And thank you for having me here today.
Thanks, Nara. I'm going to just finish here and just sort of summarize. And then also, I just wanted to kind of end by discussing sort of some lessons that we've learned about this process, particularly from the Undiagnosed Diseases Network and from running the Model Organism Screening Center. So in the UDN, we've had the privilege to sort of be involved in this study. So this is showing kind of the phase 1, where that happened from September 2015 to August 2018, where about 2,000 applicants applied, about 1,000 were accepted. You can see about 800 probands were sequenced, and we had a number of variants and genes submitted for model organism input for diagnosis. And in that first phase, we used a lot of model organism databases to return sort of key information from model organisms that may not have been looked at in the initial sequencing analysis. And that helped us narrow down amongst the genes submitted to us to about 73 that we prioritize for functional studies across fly and fish. And in red, it shows sort of gene discoveries, the categories Nara was going over, where the gene was not known to be associated with human disease. And then the black is showing genes with potential phenotypic expansion. And so the lessons were that the first lesson was sort of that good informatics and attention to nomenclature is an important aspect of these types of studies across organisms. And then the second idea was that the functional studies really can help facilitate the diagnosis for undiagnosed patients. So in phase 2, the effort was actually expanded to include additional teams, where basically now the MOSC includes a fly group, 2 zebrafish groups, and a worm group across 3 different institutions. And the key kind of final point is that the way we function with the clinicians in the UDN, the genetic counselors, the human genomic researchers, and the model organism labs is through a process of communication and sustained collaboration. So we get submissions from the sites, we perform informatics, we're very transparent about the way that we think about the genes and whether they're good candidates in a particular model organism. We speak to each other across the 3 model organisms about what's the best model organism to study this gene candidate for this particular question. And then after studies, we return information that we hope informs the diagnosis. And what we find is also by having the clinicians present patients directly to us, we actually are better informed as researchers about what pathways we're trying to study, what genes we're, what the question is about the genes that we're asked to study. And so all these communication elements are really key for using these tools, even though the tools can rapidly bring you towards, can rapidly bring teams together, the continuous communication becomes key. So anyway, that brings us to the end of our presentation. I also wanted to acknowledge our teams, and I wanted to thank Nara, my partner in this presentation, and I think we can take questions.
So for questions, I suggest that you share your screen or you show your face and you raise your hands. Both are options. And we can look at these questions. There have been a few questions that have been answered by Shinya. but I'm not sure if everyone was involved. It looks like everyone was involved.
Yeah, and I did appreciate the question about the gene nomenclature, which, you know, in my experience has been non-trivial. For human genes, we rely on the HGNC, the Human Gene Nomenclature Committee. We put a lot of attention into the use of HGVS coordinates for the variants. And then, yeah, as Shinya mentioned, DIOPT is a very key tool for finding the model organism gene and linking directly to the model organism database for the official gene symbols.
Just to add to that, in the Matchmaker exchange group where we exchange the information from one database to the other, actually we chose to exchange based on the ensemble ID because we thought it was the most reliable ID for the gene to be exchanged across different databases. So it doesn't matter how you receive it, you transform it to an ensemble ID and then you exchange among the other databases.
Any questions? I would like to say that you know, this will be publicly available and posted on through the Genetic Society of America. It may take a couple of days, but all the information will be publicly available and shared on YouTube. And so if there are any questions, you can email Michael, Shinya, or Nara, and we can then move on from there. I see no questions pop up, so
there was one, maybe this was actually, I got it as a direct message, but this probably is for a public question. Out of curiosity, how do you resolve issue of authorship?
So, so that is actually something that I think data sharing has made a lot of people change about it. People became much less worried about it at the end and more worried about actually solving the case for the patient. You sometimes just have to give up on it. You know, like depends a lot on who is doing most of the work on the paper, most of the work on the functional part, who has the most number of patients. I have not had any bad collaboration. So far, everybody was really understandable. It's multiple first authors, multiple last authors. So it became a conversation, but actually you see the change. At the beginning, people would say like, hi, we matched. What's the phenotype of your patient? And period. Now people say like, hi, we matched, my patient has this variant, that phenotype. You know, they are much more open to just share the information and less worried about being scooped. I think most of them have been collaborating and had good experiences and have no reason to hold back on any information. But it has been fine, but it's always depend on who is talking, who you're talking to, and depend a lot on who is doing what in the collaboration.
yeah, we share the same experiences with the Model Organism Screening Center. We've collaborated about the entire world now, and rarely, if ever, had an issue. We have had some issues, but you know, they've all been settled with time.
Yeah, thankfully, I have not had an issue.
John Uber is raising his hand. Go ahead and ask a question, John.
Yeah, hello, and thank you, Ugo, for organizing this series and for today's speakers. I work on Drosophila only so far, and I've been curious and in part prompted by Ugo's recent blog on the GSA to look at the human side. And it seems like most of the genes that Ugo mentions cause neurodegeneration, but is this being expanded to other diseases that cause, like, I don't know, performance problems, like, I don't know, attention deficit disorders or autisms or things like that? Or is that too complicated to automate?
No, I would say that the so for the Model Organism Screening Center, the this comes up. So in those meetings I was mentioning where there's a back and forth between the clinician and the model organism labs, this comes up all the time, and some some issues for consideration. One would be, are we using the model to understand the phenotype itself? Like, are we expecting the model to have the disease phenotype, or are we interested in other phenotypes if they inform us about genetic, sort of what's gain of function, what's loss of function? Maybe the fly wing is a great tool to understand gain and loss of function for human, heart disease or gastrointestinal disease that doesn't, on the face of it, have anything to do with that phenotype. Then, another consideration is when we're bringing up what organism is, for instance, maybe a loss of function of zebrafish that would recapitulate the phenotype might help us solve the case. So, so the idea is, first decide what the goal is of the model organism study, and then, and then sort of go from there. And I think that the other point I would make is that the UDN does have a lot of neurological phenotypes. So a lot of undiagnosed patients have either primary or kind of as part of the phenotype neurological disease. So I think there is that bias coming from the input as well.
Also, I would like to mention briefly, most genes are pleiotropic. And so when you knock out a gene in flies, you may see a variety of phenotypes in different organs. And Michael was referring to the wing, but obviously this refers to the entire fly. And you can dissect the molecular mechanisms in flies much, much faster. And you can assess if mutations are dominant or dominant negative or gain of function. And if they affect the protein function and you see an obvious phenotype, you can very quickly relate that to the gene function. And in humans, most of these genes also are pleiotropic. In fact, this is one of the problems that so many of the diseases are pleiotropic that none of the NIH institutes is interested in supporting diseases research that is pleiotropic because they're specialized in very specific organs, and that creates sometimes a problem. And
the one institute that would cover it has the least amount of money to give out. It's NIHSG, so it's complicated, but yes.
Does that answer your question, John?
Yes, very much. I was just wondering in the input side if clinicians were also entering things that at least superficially show a very specific neurological behavioral defect. And it sounds like possibly. Yes.
There are mutations that cause epilepsy. And when we knock them out in flies, we see no phenotype, but they're susceptible to seizures. They're bank sensitive they get seizures when you put them at a higher temperature. Yeah. And that's about the only phenotype sometimes that we see. So, you can draw parallels pretty easily for some genes. Sometimes it's more difficult, but many, as I said, affect different organs, and therefore one doesn't have to worry too much about it.
Thank you.
All right, we're already 5 minutes over time. If you have any questions, you can email Michael, Nara, or Shinya with respect to ModelMatcher, and we can move on. We'll have another meeting in about a month. Thank you.
Thank you for inviting me. Thank you, everybody. Bye.